All right, so thank you everyone for coming. I'm Yvette Panisowicz. For many of you, you may not have seen my face, but you've been getting a lot of emails about this. So I want to thank you all for coming to support James through his presentation and listening to all the good things that he has done. And I'm going to turn it over now to Ms. Gonzalez, and we're going to get started. So please feel free to go ahead and get yourself refreshments and whatnot, and they'll be here afterwards as well.
Welcome, everybody. My name is Alexa Gonzalez. I am a school counselor at Lawrence High School. I've had the privilege of being James' school counselor for the last 4 years, and his brothers before that, so I've done. So I first met James as an 8th grader, and at that time he had come to talk with us. I think Mr. Davis was there as well with his dad, and just to see if Lawrence High School was going to be a good place for him to go through his high school years. And I'm just so pleased that the family chose to stay here at Lawrence High School, because James has proved to be such an asset to the school and has done just amazing things while he's been here. A traditional high school program is not necessarily complete for a kid like James, who has a lot of potential and has a lot of aspirations, and we were able to accommodate, and create a program for him where he was able to really excel and push forward. So he sees himself as a man of science, but James has this really highly developed ability to synthesize and analyze information, and he is a favorite here amongst the entire staff, not just the math and science folk, but also our humanities staff. And I just want to read a couple of things that Mrs. Herzog had to say about James to kind of illustrate, the type of student that he is. James Gow is one of the funniest, most intellectually capable, spontaneous, and insightful students whom I have taught in my nearly 4 decades in the classroom. To say that having James in the classroom alters the intellectual dynamic and tone of the space would be an understatement, to say the least. His mind works on one speed, fast forward, which propels class discussions to new depths and down uncharted avenues. So James has this energy about him that he takes to every single place that he touches here at the high school and beyond. So an example of his energy is what he has undertaken his senior year, because it's exhausting for me to even talk about it, let alone live it. I'm sure parents, you're tired as well because you've been part of this. But this year, James is here for a half day. He takes 4 classes here at Lawrence High School. He's enrolled in the 2 additional advanced placement courses that he completes on his own independently through our online courses. He goes to Princeton University, where he took 1 course the first semester and then a 2nd course this semester. So he has that to accomplish. And then he goes to Rutgers to complete his independent research with Ms. Alexa Murray and also Dr. Laskin. And this on top of being a leader in Model UN and Model Congress and Science Olympiads and captain of the ice hockey team and all the other things that he's involved in within the school. So he's just an amazing, amazing young man. He's been working on this research since the 8th grade, I believe. So this has been an ongoing project, very long term. He's really been able to delve into it this year. He's been involved in the international competitive science fair circuit for years as well. So he's very honed in his presentation skills due to those experiences. So I'm just really pleased that he's been able to put this together for us so that we can also share in the work that he's done. So I would try to explain what he has researched for the last 8 years, but I don't know if I'm going to be very well versed in that. But his research topic has been using anti-tumor necrosis factor alpha as a treatment in chemotherapeutic-induced fibrosis. And I didn't practice that, so I'm impressed that it came out that so with no further ado, here's James Gow, and allow him to wow you with his brilliance.
Okay, so yeah, as Ms. Gonzalez said, the project is on tumor necrosis factor alpha with chemotherapeutic-induced fibrosis, and we'll get a little into more into what that means as we go through. But first, I want to come to sort of the start of this project. As Ms. Gonzalez said, it's been very long-term for me. It's been something I've been interested in for a long time, and I'm happy to have this passion for such a long period. So sort of the people in my life that helped me get started with this, I think it really started in 3rd grade with Mrs. Rooney. We had a sort of open-ended science project, and it was just supposed to be a 5-minute presentation on something that you found you really had a passion for in science. And this diagram to the right here, the hematopoietic stem cell and its specialization into all these different types of blood cells was the topic which I picked. The hematopoietic stem cell really fascinated me, and I really got down and I did a presentation on how it becomes the macrophage. Following that, in my 5th grade summer, I asked my dad if I could go and do some research on the macrophage. And so from 5th to 8th grade, I worked with one of his colleagues, Dr. Shengjian Guo, and I learned about biochemistry of cell signaling, how the macrophage differentiates, and sort of the surface markers that are affected during that process. And then in 8th grade, I applied for the summer after, I applied for the Liberty Science Center program, where they partner you with a researcher in the field, usually in New Jersey or Pennsylvania. And that's when I started working with Dr. Laskin and her graduate student at the time, Alessandro Venosa, who is now a doctor. And following that, when he graduated, I started working with Alexa. And so sort of through here, I've moved from an in vitro study of macrophages and looked at now in vivo models and 2 different chemotherapeutics and how they affect the pulmonary tissue and the role that the macrophage plays in that. So a little bit about what the macrophage is and how it works in an in vivo model. There are 2 distinct populations of macrophages. The first is in the vasculature, and so the hematopoietic stem cell populates the bone marrow. And within that bone marrow, it matures all the way to the monocyte, and then it goes through the vasculature when CCL2 is released by a tissue in stress, and then it translocates to that tissue. CD11B is the integrin that's expressed by these monocytes or macrophages after they've been recruited, and it's necessary for them to penetrate the basement membrane. And in smaller expressions, lower expressions, it's needed to move throughout the parenchymal space. So like I was saying, CD11B in high expression, that's what's needed to penetrate the basement membrane and get into the parenchymal space. In lower expression, that's what's used to move throughout this parenchymal space and the interstitium. The second population of macrophages we call the resident cells. These are yolk sac-derived cells, so they were never hematopoietic stem cells. And then within the lung, they're always there, whether we're injured or not, whether there's an insult. And they're more biased to an anti-inflammatory pro-repair phenotype, and rather than the monocytes, which have the ability to adapt either phenotype. So looking at the macrophage phenotypes, initially when people started researching macrophages, we said there's just an inactive and an active macrophage. And as they did more and more research, what they discovered is, well, there's such a disparate profile of macrophages. Let's segregate it into 2 groups, M1 and M2. As research has continued today, we've sort of realized that you can't really segregate it into 2 populations. It's more like a spectrum, and every cell can be in a different place within that spectrum, but for simplicity's sake, we still keep it as this M1, M2 profile. M1 cells, they're cytotoxic, they're pro-inflammatory. We call them classically activated, and one of their main activators is tumor necrosis factor alpha, which obviously we'll talk a little bit more about later. The alternative activation, or the M2, is an immune suppressor. It plays a huge role in tissue repair and sort of fungal recognition, and it uses the other elements of the adaptive immunity to respond to issues. So nitrogen mustard, right? What I first started working with was the cytotoxic agent nitrogen mustard. A little bit about why to start. It's a heavy analog of sulfur mustard. So when we think of mustard gas that was used in the World War, it's sulfur mustard. It's gaseous at room temperature. Nitrogen mustard has this additional CH3, or it's actually an R chain. It can be variable depending on the nitrogen mustard, and that causes it to be heavy enough to be liquid at room temperature, and so it's more easily used in the lab because it's just a little bit safer. The second reason that we use nitrogen mustard is it's actually still quite a common chemotherapeutic. Usually we use a cocktail of 3 drugs, of which nitrogen mustard is often used for lung cancer. It's a very aggressive chemotherapeutic, and that's sort of its limitation of use. And it causes, in the long term, a lot of the injuries which we'll talk about today, and that's why we have to limit its use. So if we could potentially solve the issues of the chemotherapeutic, it would actually make for a better treatment. Well, what does it do as an injury? It causes methylation of the deoxyribose sugar chain, and that causes basically random metastasis throughout the genetic code. So these random morphogens in the genetic code causes differential expression, and that causes the cells to possibly take on an inappropriate response to an injury. It's a very strong pro-inflammatory response from the body that is sort of unresolving. Normally when we think about an injury, we think about an innate response at about 3 days, and that sort of fades into resolution. But with nitrogen mustard, because of its oxidative stress that it puts on, and because of this random mutagenesis, it causes an unresolving pro-inflammatory response and a lot of cellular recruitment. And then finally, at about 3 to 4 weeks, we get this fibrotic injury, and this is sort of the long time point of the injury of nitrogen mustard. And it's something that we observe in a lot of patients who've received nitrogen mustard as a chemotherapeutic. So the goals. The goals of my research were to find a solution to this chemotherapeutic-induced fibrosis, determine if that solution could potentially be a drug that's already in circulation, and determine if the inflammation is mediating a fibrosis or they're 2 separate events, and if the solution that I provide, the drug that I'm using, would affect the inflammatory stage and in turn affect fibrosis or just directly affect fibrosis. And then what is the macrophage's role in all this? We know it's a big immunoregulator. It helps the body decide innate or adaptive immunity and when to make that transition and how. And so investigating its role will be key to understanding how we can actually find a solution. So the initial choice of treatment was valproic acid. Valproic acid is an epigenetic inhibitor. So a little bit about epigenetics. Normally we have this very cyclic epigenetic profile where we have a relaxed chromatin structure where there's acyl groups. They actually cause more of the unwinding of the DNA, so transcription factors are more readily able to get to the DNA wrapped around the histones. Then we have these things called HDACs, or histone deacetylases. When they remove this acyl group, we get a condensed chromatin structure. And then acetylases add it back, and so on and so forth. So we cyclically have this relaxed or condensed chromatin, and this is the way in which we naturally have epigenetics is actually hereditary. But valproic acid removes all of these HDACs, halts this cycle suddenly, and causes acetylation and permanently relaxed structure. Why would this be potentially useful in this particular application? Because we know this random mutagenesis is changing transcription in these cells. And so possibly by having such an effect with valproic acid, we could change the transcription in a way that will positively affect the inflammation. So experimental design, the 8-week-old rats, they were given nitrogen mustard intratracheally just through one singular injection. And then 30 minutes following, they were given another intratracheal dose, this time of their treatment, which would either be a control, which would just be a phosphate buffered saline, or the valproate or valproic acid. And then we would take a number of different time points to analyze peak of inflammation, how inflammation transfers to resolution, and then the fibrotic time point, which is 28 days. So a little bit about histology, because this is going to come up a lot during the presentation. It's a way that we really analyze the damage that occurs to the lung. This here presented is just a control hematoxylin and eosin stain. Hematoxylin stains the nuclei and calcified cell structures violet, and eosin stains the cytoplasm red. And sort of it gives us an idea of where the cells are located, how much cellular infiltration occurs, if the septal damage has occurred, and if there's any epithelial thickening. And so sort of as an example, this is a control completely untreated. You can see it's sort of very jagged septal edges, so there's a high surface area. There's not a lot of cellular infiltration, and the epithelial lining is not thick at all. At 3 days, this is following nitrogen mustard treatment. You can just see that there's a plethora of cells here. There's a lot of cellular infiltration. There's definitely some epithelial thickening, and there's certainly less septal definition. And at 7 days, that cellular infiltration starts to fade away, but we still see a lot of damage in the lung. So proof of concept, right? We know what valproic acid is supposed to do. It's supposed to remove those HDACs and cause that relaxed chromatin structure, which would change the transcriptional profile. but we had to prove that it was actually doing that. I mean, we don't know if it would work particularly in this in vivo model, and we didn't know how nitrogen mustard could possibly affect this HDAC effect. So we did immunohistochemistry for histone deacetylase 2. It's actually kind of hard to see on this screen, but brown color is the presence of that protein done on a thin section of lung. So we add a primary antibody and a reagent. When they react, it produces a brown color where that protein is located. So you can see that there's a very faint brown in the macrophage stain of 3 days, and at 7 days, those macrophages are a much darker brown. And it's there's very little, if any, noticeable brown in our valproic acid. So basically what we can say is definitively valproic acid has inhibited the presence of histone deacetylase 2. Also what we can see is that histone deacetylase 2 is nitrogen mustard dependent, meaning that at the control there's very little, if any, histone deacetylase 2. At 3 days there's significantly more, and at 7 days significantly more than that. So inhibiting this certainly will have a noticeable effect on nitrogen mustard injury. So then next we're going to look at gene expression. So we know that valproic acid is having our desired effect, but how is it having this? What is this effect having on the macrophage response? Right. So we do a process called reverse transcriptase PCR, where we've lavaged the lungs with that phosphate buffered saline. We draw that liquid back up, and all of the cells that have now been picked up by this phosphate buffered saline, we isolate the RNA from those cells. We turn it back into DNA, and then we use Taq polymerase, which is like a high temperature polymerase, to replicate that DNA until there's noticeable amounts. The graph is presented in fold change, so it's a very sort of arbitrary amount of RNA when we do the measure. So the way we present it is by saying, well, what's the change in the treatment group over the change in the vehicle group, and then how does that fold change over a housekeeping gene? All that really means is sort of how is this particular gene expressed versus a gene that's pretty much constant all of the time. And that's called the 2 to the minus delta delta CT method. But what's important to note is we can see that in the red, I've sort of outlined the pro-inflammatory genes that have been downregulated by valproic acid. And in the blue, I've shown that there's an upregulation of anti-inflammatory genes, also at the 3-day time point. A little bit about the genes that we're looking at. INOS is downstream of the NF-kappa B transcription factor. It plays a big role in changing the role of other proteins by nitrosylating them. So not only is it pro-inflammatory itself, but it changes the way that other proteins act within these macrophages. Also, from the NF-kB pathway, a cell can only express INOS or arginine, and if it expresses INOS, it takes on this pro-inflammatory function, but its lack of arginine will eventually cause it to apoptose, and so that's the normal pathway, and it's something that we'll see go wrong as a result of nitrogen mustard. COX-2 is prostaglandin synthase, and it's just another pro-inflammatory hormonal regulator. IL-10 and ATR1, IL-10 plays a role in lipid digestion, and ATR1 in epithelial repair, but what's really important to take away is that we're downregulating pro-inflammatory genes and we're upregulating anti-inflammatory genes. So we know that there's some change on the genetic level, on the transcriptional level, but how is that being translated? Is there another change at the translational level, or would we see the same results? So this is immunohistochemistry. It's, again, histology, same idea as the HDAC, but this time with the proteins INOS and CD68. INOS, like we just talked about, is this pro-inflammatory gene. CD68 is a marker of maturity. It binds to low-density lipoprotein. And it plays a role in macrophage phagocytosis, but again, it's sort of associated with anti-inflammatory. So what we see is more anti-inflammatory expression, and we see less pro-inflammatory expression with valproic acid, because this is our control line, our valproic acid, and same here. So it sort of matches exactly our gene profile with our protein, that there's less pro-inflammatory, more anti-inflammatory. So now that we've looked at sort of the population, we've looked at the whole lung, we wanted to take it to a cell level. So all the way on the left-hand side, that's the resident population, which is very unchanging, which is sort of to be expected. The resident cells are there all the time, and whilst they're activated by injury, they're already present. They're not really changing their recruitment profile. In the middle, we have our immature infiltrating cells. These are the cells that have recently just become macrophages or are still monocytes. So they're very young in their recruitment. They're definitely not activated yet, but they're CD11b positive, which means they're moving. So they're probably moving through the vascular, just entered through the basement membrane, or perhaps they're moving around within the tissue itself. At the end, we have these mature cells, and this can either be an activated resident cell, now that it's expressing CD11b, which is that movement integrin we talked about, but it's CD43 negative. And what that means is it's been a macrophage for a significant period of time, or it's received a strong activating signal. And so, well, what does this really mean in the context of the fact that we've seen downregulation of pro-inflammatory and upregulation of anti-inflammatory genes? What's likely happening, and what I would hypothesize is happening from this data, is that there's such a strong pro-inflammatory signal as a result of the nitrogen mustard treatment without our valproic acid that it's causing cells to actually be activated, hence this mature population, before they even reach the lung. They're already receiving activating stimuli. The reason they'll still be immature when they get there as a result of valproic acid is because we're dampening this pro-inflammatory signal. So the ultimate question, though, is the animal healthy? Have we actually made a positive impact on this animal's fibrosis as a result of nitrogen mustard? The answer is not really. There's a very small change. We see a lot of these transcriptional changes and protein expressional changes, but there's still, you can barely see a difference between the 3 days and the 7 days. There's maybe less cellular infiltration, but when scored these slides, they came out very similarly between valproic acid and the vehicle. So what does that mean? Well, it means we need to move on. We need to try a new treatment. We've learned a lot about how this process occurs, but we haven't found the solution with valproic acid. So, what are the issues with valproic acid? First of all, obviously that H&E stain that I just showed you shows there was not much of a difference. The health of the rats based on their body weight was still pretty much the same. There was no significant change in that trend. And also, valproic acid is what we call sort of a very dirty drug. It doesn't have a very specific target. Although we know that it affects HDAC, it could be affecting other things that we're not really sure about. And that makes it sort of difficult to analyze. So what we can take away from that, though, is a few new hypotheses that I've been able to form based on the data that I've already presented. The first of all is you might have seen a trend with sort of a lipid regulators. The CD68, that's a lipid regulator. The COX-2, that plays a role with lipids. So all these things that are related to lipids, maybe that has something to do with our injury. Also, what we saw, a lot of these changes were at 3 and 7 days. So that means this is still the inflammatory stage. And probably to make a significant impact, I would personally hypothesize that we need to target that inflammatory stage. So the next choice that I made was tumor necrosis factor alpha. That's what was next used in this project as a potential co-chemotherapeutic or co-treatment to this nitrogen mustard. And why would we select this? Well, first of all, Remicade is an anti-TNF inhibitor, so that is already on the market. And so we know that this type of thing is already used, this type of drug is already used, and so that sort of fulfills our initial goal of can we find something that's already on the market. It also, tumor necrosis factor alpha plays a key role in the activation of pro-inflammatory macrophages, like we talked about all the way back on that first slide, through the NF-kappa B pathway, which we know regulates INOS, that we also saw a change in with valproic acid. The other thing that we saw is there is maybe an issue with apoptosis. There may be some cells that are actually vacuole, causing all that lipid to become vacuoles and not able to apoptose as they should. And so we also know TNF-alpha plays a role in apoptosis. So perhaps by changing this, we could resolve the cells that should apoptose but actually do not. So the experimental design, very much the same, except instead of valproate as our treatment, we're going to use an anti-TNF inhibitor. Also, this will not be given in an intratracheal dose. It'll be given intravenously through the tail vein. So anti-TNF injury in nitrogen mustard, first important question because does it actually work? We saw that there wasn't much of a change last time, but we wanted to make sure that there would be a change, that there was a positive change in the health of the animal, and this time. It's very difficult to see on the screen actually, but what we can look at, we're looking at the same sort of idea of cellular infiltration, septal definition, and epithelial thickening. I think the easiest way to point it out is at 3 days, you can certainly see some regions that have much less cell infiltration with TNF as opposed to the PBS. At 7 days, you see a much thicker epithelium with our PBS rather than our anti-TNF. And then we can also see that there's a little more septal definition at this 7-day time point. So we do significantly see a change here. When these were scored by people who didn't know what the treatment was, they did receive different scores. So the next question is, we repeated all of those results that I showed you about INOS, COX-2, the gene expression, and the protein expression. But for time's sake, I won't show you all those again for anti-TNF. But we talked about the idea that phospholipids could possibly be involved in these cells that are not apoptosing. They could be regulating the consistent and prolific inflammation that we know is not good for the pulmonary tissue. So first we want to say, okay, if phospholipids are involved, then they must be changing somehow. And so what we just took is a nitrogen mustard slide. There's no TNF involved here, but we just took some macrophages and we took a piece of lung tissue and we stained it with oil red O. Oil red O just stains phospholipids, so we wanted to see, is there any change? With the control, without any treatment, very little stained phospholipid relative to the amount that we can see with nitrogen mustard. So we know that nitrogen mustard is significantly changing phospholipid presence. So why would phospholipid be important in the pulmonary tissue, and why is it something that we would be interested in studying? Basically, in order to breathe appropriately, you need to have a surface tension be able to be controlled by SPB and SPC in the lung. And basically what SPB and SPC do is they catalyze the change from phospholipid being in a monolayer around the pulmonary surface, and then also being in a micelle. And so micelle is just sort of a packed ball, and it's sort of, this is the way that SPB regulates the surface tension, such that at any given time we're able to use all of our alveoli. Because if you had constant surface tension and you weren't able to change this based on the size of the alveolus, what would happen is the pressure would be much greater in the large alveolus and effectively zero in the small alveolus, and that would cause your lung to collapse. So, SPB phospholipid ratio, how did it change, and what does it mean? How can we interpret this data? The first thing I would point out is, based on the error bars, there's actually no statistical significance between any of these white bars, any of the anti-TNF bars, which is a really encouraging sign. Essentially, we want this SPB to phospholipid ratio to be constant all of the time, whether we're injured or whether we're healthy, and it's necessary in order to maintain that surface tension that we talked about. So the fact that anti-TNF can restore this effect such that it has the same with control and with injury is a very encouraging sign. We also see is the nitrogen mustard alone, without the anti-TNF treatment, it causes a serious spike in SPB initially and a serious drop in SPB at the end, or spike in phospholipid, because the presented here is the ratio. What does this really mean? Well, at 3-day time point, probably this spike in SPB to phospholipid is due to the fact that the epithelial cells, even 72 hours following injury, are still under a lot of stress, and so they're going through a lot of transcription in order to try to resolve that stress. Additionally, the macrophages will possibly be actually phagocytosing this lipid. At 28-day time point, however, on the contrary, there are a number of potential solutions for the reason why the phospholipid would be so high, but possibly because the presence of all of these macrophages that have started to digest but were unable to complete the digestion of such phospholipids. And that might be likely due to the fact that they could be oxidized from the oxidative stress of the nitrogen mustard. So SPB or SPD, only one can be made by an epithelial cell at any given time. So they're exclusively transcribed one of the other, but we see sort of here at 3 days SPB is much higher without our anti-TNF treatment, and we also saw SPB was higher without our anti-TNF treatment. So this seems sort of impossible. If you can only transcribe one or the other, how are they both high? And well, what this sort of is indicative of is an extreme amount of stress, such that there's just more total transcription. Also, the fact that SPB maintains its high levels, SPD through 7 days, shows that they're still under stress even 7 days following an injury. They only get one dose, so it's not like we're replicating this injury, but they're clearly not coming to any sort of resolution. And at 7 days, we should really be expecting a transition from inflammation to resolution. And this really just shows SPD is an immunoregulator. It can be pro or anti-inflammatory, but its transcription is sort of indicative of cell stress. So by reducing the stress of our alveolar cells, it's very indicative of a more healthy lung. So future. This is sort of a sidebar that I included because I think it's very interesting for the future of this research. Basically, the lung was sectioned, the entire lung was sectioned, and then CT scanned. And there is a video of this. Unfortunately, I don't have it, but basically, you can see that this is a 3D model. It's not just the 2D sections of lung that we've previously been working with histologically. And the red represents where air is, the blue, inflammation and liquid in the lung. And so the value of such a CT scan would be, first of all, we can reduce the amount of animals that we can use, and second, reproducibility. Whenever I was showing those histology slides, I had to show you 2 different sections of lung because I can only stain the section of lung once. And so what this would actually enable us to do would be to more reproducibly do the same set of analytics on the same section of lung. And obviously, most importantly, I think, is that it gives us a 3D structural understanding of the lung rather than just looking at 2D protein expression. So after that whole analysis of phospholipid, I sort of moved on one step further. We'd gotten pretty far with the anti-TNF in nitrogen mustard, and we understood that it made a positive effect. It changed protein transcription, it changed translation, and it changed effect on a cellular level. But we still had a lot of work left to do, being that this was only proof in one animal with one cytotoxic agent. And when working in rats, you're sort of heavily limited by the fact that not a lot of people work in rats, and so there are not a lot of techniques, there are not a lot of primary antibodies, and that is actually a huge limitation that people have encountered in the field. So this time we moved to a second animal, being a mouse, and we used a new cytotoxic agent, which is bleomycin. This is a chemotherapeutic for breast cancer generally. And so the advantages, obviously, being that if we can prove it works in two cytotoxic agents in two animals, that would be really huge. And also, there are way more complex techniques that are already developed for mice rather than rats, and so that enables us to do some new research. There's also more literature, so there's a better understanding of what we would be looking at. So the experimental design, like I go through this, bleomycin would be 3 units per kilogram, and the unit is sort of very arbitrary in how much it would take to kill an insect. But the point is that we changed what the cytotoxic agent was, and we're still using the anti-TNF treatment because we believe that this has the potential to be an effective co-chemotherapeutic. So again, we went through that phospholipid data that I presented, and we replicated that. We went through the protein expression, replicated that, and we went through the gene expression, and we're starting to replicate that as well. But what I'm going to show to you, just for time's sake, is the new piece of research that we were able to do in this bleomycin mouse model, and that's lung digestion. Effectively, normally when we analyze the cells in the lung, we just do that bronchoalveolar lavage I was talking about, where you push PBS in, you pull it back out, and then you analyze the cells that come out with that liquid. In this technique, we actually remove the lung, we digest it with collagenase so that all of the collagen in the lung sort of releases the cells that were formerly stuck to it into the interstitium. Then we force it through a cell strainer, and now we basically have all of the cells that are from the interstitial population. And this is a population that's previously not been examined within the context of this injury. And so after that, we wanted to say, well, we really care about the macrophage population. And so how are we going to just analyze the macrophages? We have this whole population of interstitial cells. So what we actually did is we added a primary antibody for F480, and then we added a magnetic bead that binds to that F480. And so then we basically place these test tubes in a giant magnet, and then that magnet causes the beads to move to the side of the test tube. We can aspirate all the other cells from the solution, and then now we have a purely macrophage population. So what is presented here? Well, this is flow cytometry data. It's when we run that cell level with valproic acid, it's the same sort of analysis, presented in a different way. And so we did it with 4 different primary antibodies. The first is mannose receptor. Mannose receptor is just a very classically accepted M2 activator. Then LY6C, which is in the red in the third column here, that's a very pro-inflammatory cytokine. We don't really understand a ton about what it does, but we know it's only expressed where there's where there's actually inflammation itself. And then the other 2 are the 2 integrins we previously talked about, CD11C, which is only expressed after a significant period of time in the lung, and then CD11B, which is needed in high levels to penetrate the basal membrane and low levels to move around the parenchymal space. So we know that anti-TNF is this. It's supposed to reduce inflammation. That's what it does by inhibiting the NF-kB pathway. But we actually saw exactly the opposite result when we looked at this lung digestion. We saw downregulation of anti-inflammatory, which is that mannose receptor, and upregulation of the pro-inflammatory LY6C. Also, we saw more of the cells would be CD11C positive, which means in the nitrogen mustard alone, rather than the anti-TNF, which means there's actually more recruitment as a result of this anti-TNF treatment. So how can we explain this complete converse effect of what we were expecting with anti-TNF? Well, actually, it took a while to come up with this, but what we realized is that it's an anti-TNF antibody that's being added. It's not actually inhibiting completely the expression of anti-TNF. Essentially, what I would hypothesize is that these cells are taking on a pro-inflammatory phenotype, but then they're not able to perform their function as a result of this antibody. And so then they're actually recruiting more and more pro-inflammatory cells, but none of them are having this phagocytic pro-inflammatory effect. And so that actually causes none of the cells, or a much smaller percentage of the cells, to be actually uptaking this oxidized phospholipid, and that might be improving the persistent inflammation that we'll see. What sort of suggests that this hypothesis is correct is at 21 days, we actually see less pro-inflammatory cytokine, being that LY6C. So we still see a lot of pro-inflammatory expression with nitrogen mustard alone, but this anti-TNF treatment stops the persistence of the inflammation, which is one of the key issues that we notice with bleomycin injury. So the next analytic that we did was cell count. This is just purely taking that BAL that I talked about and just seeing how many cells are present and what does that mean. So we would normally sort of hope that there would be a lower cell count at 21 days, right? If resolution was more complete, then we should have less infiltration. But a few things that we notice that sort of explain this higher cell count are, first, on this slide, in the yellow, it may be a little hard to see, but in this negative column, we can actually see at 21 days, much higher percentages of the cells are not CD11b positive at all in the TNF rather than the PBS. And cells that are not expressing any CD11b can't move. So they're very unlikely to be activated. They're very unlikely to have of much an effect on the injury. And so possibly one explanation for this is that we have actually more inactive cells, which is suggesting a better resolution. And the other thing is what comprises this population of cells. And when we did a cytospin where we essentially take that BAL and we put the cells onto a slide and then we can sort of look at them under a microscope and understand what type of cells they are, we actually see less fibroblasts, which do the collagen deposition that makes fibrosis. And also, we see less foamy macrophages. And the foamy macrophages are what we call these macrophages that have begun to digest but can't completely digest this oxidized lipid. And the way we can identify them is they're much, much larger and they're very vacuoled. And their presence increasing over the time course suggests that they may be playing a key role in this persistent inflammation. So by seeing less of those, that's also another very encouraging sign of reducing this persistent inflammation. And also, these less fibroblasts. Fibroblasts usually can possibly come from a monocyte, and so these monocytes apoptosing appropriately is possibly why we have less fibroblasts, which again would be an encouraging sign for the presence of fibrosis. So, to the most important thing, does it work? Are the animals more healthy? Are these mice actually being improved by this anti-TNF treatment? This is the change in weight from when they began to when they were put down. Essentially, they're receiving a chemotherapeutic at 7 days, so we would expect weight loss, and that's what we observed, generally speaking. Very variable. Each animal responds differently, but we do see a significant change in the negative direction as a result of the treatment with bleomycin. But what we look at 21 days is there's actually weight gain, significant weight gain from the bleomycin with TNF, as opposed to the animals that were just given the saline treatment. They're actually still losing weight. And this is a very encouraging trend because these are 8-week-old mice. Gaining weight is the normal health trend for them. And so they're essentially returned to more normalcy than those that didn't have our treatment group. So the last measure of the health of the lung is an H&E stain at 7 days and a trichrome stain at 21 days. The reason we picked these particular stains is hematoxin and eosin is great to understand inflammation, and trichrome is great to understand collagen deposition or fibrosis. So at the acute time point, one concern you would have based on that lung digestion data is, well, there's a lot more infiltration. There's a lot more cells present. That could possibly facilitate a stronger inflammatory profile that could actually cause a serious injury to the lung. But what we noticed is that while cellular infiltration is possibly higher or effectively the same with anti-TNF and bleomycin alone, the actual score of the histology based on infiltration, septal damage, and epithelial thickening, again, is pretty much the same. The median is exactly the same. And actually the range is lower, which means less of these animals are severely inflamed. And that's actually really an encouraging sign, but not very significant. They're essentially the same. But what's important to understand is at 21 days, what we're really trying to do is change fibrosis. So if we change fibrosis at 21 days and the animal is able to recover from the inflammation, then that's a significantly healthier lung than with nitrogen mustard alone. Bleomycin alone in this case. So the bleomycin alone, you can see a lot of these heavily blue regions. That's collagen deposition. And this is a actually very low amount of collagen deposited in the anti-TNF. And I think even for people that don't see a ton of histology, people can still see that there's a significant change in the amount of blue that's present, and that's really encouraging for the amount of collagen that's deposited and is pretty indicative of a healthier lung and possibly a more functional lung. So conclusions based on all of this research would be that phospholipid synthesis and regulation is really important in nitrogen mustard, and then also bleomycin-induced fibrosis, possibly because of these foamy macrophages are phagocytosing this, and then they're unable to apoptose because they can never complete their function because they can't digest oxidized phospholipid, and that's probably causing this persistent inflammation that we observe. We found that valproic acid, as of now, is not really an effective co-chemotherapeutic, but potentially it could have implications in the future if we were to change the dose or how it was given. But we did find that anti-TNF, it really did reduce this chemotherapeutic fibrosis. We can look at that collagen deposition, we can look at the health of the mice, and then it worked across 2 models. It worked in the rats and it worked in the mice. And then finally, anti-TNF, it doesn't reduce this M1 phenotype, but what it does do is reduce the pro-inflammatory effect. So they're still taking on this M1 phenotype, but since they're unable to perform that function, it's not having this pro-inflammatory effect, which is a really encouraging sign because what it causes is a more complete and healthy resolution. So where would we go next with this project? And really, by this, I mean, where will I try to go this summer? Non-inflammatory fibrosis model is really, I think, the biggest goal that I have for this summer. I still believe that the way that anti-TNF is having its effect is got to be through the way that it regulates inflammation, and that is based on the fact that significant changes are taking place at 3 and 7 days, which is the inflammatory time point. But that's certainly, the data does not prove that definitively, and some people would definitely disagree with that. And so what I want to test is a fibrosis model with anti-TNF with no inflammatory stage, and then see how that would be affected. And such a thing we could do would be amiodarone for that. I want to further investigate the phospholipids. I haven't had a chance to do the SPB in the bleomycin model, and if I could replicate that SPB to phospholipid ratio being exactly the same across time points, that would be a very meaningful piece of data that if I could prove that across 2 models, so I'd like to do that. Maybe better understand TNF's role in apoptosis, because apoptosis is certainly important in this process and in limiting this longevity of the inflammation. And then we haven't had a chance to do the RNA yet in the bleomycin model, even though we had a chance in nitrogen mustard. Okay, that was pretty much done. I think that's it. The last thing, most important, will probably be the reproducibility of the results. The number of animals that we've done is very low right now, and anti-TNF has had a few animals that have not responded. And that's caused the error bars on some of our statistical significance data to be pretty large, and that makes it hard to draw definitive conclusions. So some of this just needs to be repeated and proven that the first result was right or perhaps wrong, and then that would allow us to make more significant conclusions. So just a lot of people I have to thank. Dr. Laskin for taking me in as an 8th grade student. That was very kind for her, and I really enjoyed that experience. Now Dr. Venosa for mentoring me and starting me off with that valproic acid data, and Dr. C.J. Guo for even before that. Obviously, thank you, Alexa, for helping me this past summer. That's been a great help, and especially as an under 18 researcher, there are a lot of limitations on while the animal is alive what you can do. Charisse, Danielle, and Helen, and all of the rats at Rutgers for helping me out. And the Counteract Grant and the Liberty Science Center program for getting me there. Also, I just want to thank Ms. Panasovich for putting this together for me today, Mr. Adam and Ms. Gonzalez, and also just for allowing this to be a curriculum that I could even do at Lawrence High School. And a special shout out to Mr. Davis and Mr. Dauber and Ms. Gonzalez for that first day when I was an 8th grade student, and I wasn't even sure if I would come here, but I'm really glad that I did, so thank you very much. The last thing I would like to do is just thank my family very much, my mom and dad. None of this would be possible without you, so thank you very much. Does anyone have any questions?
It seems to me like you're a high school kid and you've done this project, but the project embedded in it is at least a full 4-year biology degree. And embedded in it is graduate level processes.
Or doctoral level.
Doctoral level. And my question is this. when you were like picking up a textbook of pathology, pathophysiology, histology, how did you dig into those other fields that you now have a conversational understanding of? When did you do it, and how did you do it? There are those other fields that you haven't had coursework supporting.
Yeah, absolutely.
That's funny, because my question was going to be, if you do this, how can you do your laundry?
I would say it really started with Dr. TJ. When I sort of first went in, I would say that first whole summer, I really didn't do any research. I didn't really touch anything as far as actually doing some science. But being with him in the lab and just actually talking through everything that he did there sort of began me on that path, I would say, to understanding the field. And then, a huge part, I think the beginning of every summer starts with reading the papers that have recently been published in that field. And I'm sure Alexa would tell you the same, like every time you start a project, you've got to just read the literature that's been recently published there. And so, every time I would encounter something I wouldn't understand, I'd just have to learn what that was in order to better understand the procedure by looking it up, looking up different research papers, and sort of just consulting wherever anyone in the lab that I could find that could sort of explain that to me. But like I say, it really started with Dr. CJ, and I think the only way that I could really keep up with that is to read the literature, read the papers. Anything else?
So James, go ahead. You talked about where you want to go next, and again, as a 18-year-old young man who certainly, obviously, very demonstrated the great knowledge that you have, what does that truly mean? I mean, are you looking to do the research on your own? You're looking, as you talked before, with now going off to college, you're looking to do that through internships? And where do you ultimately want to go?
Yeah, so as far as research on my own, I don't think research will ever be on my own. It has to be collaborative in some respect. But as far as going through, this summer, I'll be up there, health permitting, as far as the delay goes. But I'll try to finish up as much as I can based on this research. But I think as far as sort of addressing again what Mr. Davis said, I've got a very focused knowledge. It doesn't branch out with this depth to some other field. So I think heading into college, I would like to try to do similar research, probably still in the same field, but maybe branch out a little bit, improve my breadth of knowledge, and then possibly this will be what I come back to following college, but maybe not. Maybe it will be some other research. But certainly, as far as the future goes, I definitely want to do academic research.
I didn't mention James is going off to Dartmouth in the fall, so I assume you're going to need to continue your research there.
Absolutely.
Do you have any recommendations for young people wanting to get involved in science research while they're still in high school? Because it's amazing that you've accomplished this much, and I know of kids who would love to have similar opportunities, and it's hard to navigate how to even begin.
Yeah. So I would say that the Liberty Science Center program is really the way to do that. You need no prior research experience to apply. You just need to have completed your honors chemistry and honors biology at your high school, or shown your proficiency in those subjects elsewhere. Or basically, then you can apply. And actually, the opportunities are not just in this sort of immunology field. You can do physics, engineering. I mean, they have a lot of opportunities for you there. And usually people apply around their sophomore summer, but you can apply before or after, either one, and they will pair you with a researcher, and they really take it from there. I mean, you really need no prior experience. I think it's a great program, and if people from Lawrence applied, that'd be very cool. It's a little far of a drive from here, so I don't think maybe people in this area know as much about it, but I think because a lot of people at Rutgers come from that program, that's sort of how I found out about it. And I think it's a great program. You write an essay, you do an interview. If you get in, they pair you with a researcher. And usually if you want to go back and work with that researcher, if you do a good job your first year, they'll pretty certainly take you back. So I think that's a great opportunity for anyone that's interested in something like that. Anything else? Good? All right.
So I just want to thank you all again for coming, and thank you, James. We can't thank you enough for sharing this with us. Some of us who are in the science field, I don't know that I'll speak for myself, but spinning, it has some courses in cell biology, but not like that. But anyway, it was all very good. And I also want to acknowledge that James is one of the first students here in Lawrence to do this type of an option 2, or a study outside of the walls of Lawrence. And you all are the first participants in such an event by coming to hear the research that James has done and having him share this experience with us. So we charted a lot of new ground here today, and you were all in on that first level floor. And James, you've set the bar very high for students. Please do this type of presentation to this large of an audience, and you may have done this type of a presentation to audiences that better connect with your message, but you did this in front of several strangers and maybe people that you've seen along your career here in Lawrence. So I want to thank you for accepting the challenge and doing that presentation and helping us establish this for future students. So, thank you. Help yourself. Again, I can't thank you enough for coming.